Hello friends, Ace Botany, next episode. And in this episode, we'll be discussing photosynthesis, third one. And what all things we'll be discussing in this episode. For that, I will share the screen so it will be convenient. I think screen came. So we can proceed. See, almost 9 topics are given, starting with the photo, divisions of photosynthesis, photosystems or PS, light harvesting complex LSC, assimilatory power, electron transport, photophosphorylation, cyclic and non-cyclic electron transport system, ETC or ETS. Then finally, theory question and entrance question. It's almost 9 items. So we can first start with the term photo. So when we discuss the term photo, it is a familiar term for us. Photo generally means light. And in physics, there is a branch called optics. Biology side, we may use the term like photobiology, even photology. Generally deals with effects of light. For us, it is on plants. And starting with the photo as a prefix, basically 5 terms connected with this chapter. Basic term photosynthesis, then photophosphorylation, photooxidation, photorespiration, and photolysis. That is regarding first page of third item in photosynthesis. I think idea is clear. So we can proceed to next page. So when we proceed to next page, photosynthesis has got 2 segments. One is called light reaction. Other is called dark reaction, second. And next item is photosystems, otherwise called pigment systems, short form PS. Then there is one more term, PSU, photosynthetic unit. So all this will be observing. Pigment systems are also called photosystems, PS. And what is this system? See, when we discuss in a practical manner, these are groups of pigment molecules which participate in the conversion of light energy to chemical energy. And photosystems has got subdivisions, under which one by one we can observe. One is called PSU. PSU full form is photosynthetic units. And what is PSU? It is the smallest structure in a chloroplast that will absorb sunlight and emits electron. It will be given out. After absorption of sunlight energy, electrons will be emitted out. Electrons will be emitted out. A little bit deeper manner when we discuss it, that is initial light-driven charge separation of photosynthetic reactions occurs. And there are certain other terms for it. It is otherwise called quantasomes, presently not in NCERT. Previously it was there, connected with the 2 famous scientists, Park and Begins. Photosystems in green plants, PSU, occur in 2 distinct groups. And they're often called photosystem or pigment system with the number PS1, PS2. And how that numbering came, that is the order of discovery. PS1 discovered first, then PS2, just like coenzyme 1, coenzyme 2, etc. Now, on the right side, you can find NCERT diagram. It is a labeling question. Now, what are the components of PS1 and PS2? PS1, its reaction center is P700. That is, the chlorophyll molecule can absorb the wavelength of 700. Then, after reaction center, next component is light harvesting complex, LSC. Again, which has got 2 segments, LHP, light harvesting pigments. which is otherwise called antenna complex, and proteins. Protein has got a function increasing efficiency. That is regarding PS1. So PS1 is concerned with the wavelength P700. Chlorophyll will be able to absorb 700 wavelength of light. Light harvesting complex LHC has been divided into two components, LHP, light harvesting pigments, otherwise called antenna complex, and proteins. Similarly, second one is PS2. That is this one. Here you can find PS2. Again, two components, reaction center, but here remember it is P680. And light harvesting complex has got light harvesting pigments and proteins. And the proteins on both the PS1, that is concerned with the light harvesting complex LSC, is supposed to increase efficiency. I think that idea is clear with the PS1, PS2, reaction center, light harvesting complex, LSP, and proteins. I think it is clear. So we can proceed to next area where PS1 and PS2 has got many differences. Famous theory entrance question, and some of them includes, not all, many are there. Some of them include between differences between pigment system 1 or photosystem 1 and pigment system 2 or photosystem 2. Finally, we can proceed. Pigment system 1, it is P700 wavelength. Pigment system 2, P680. Place of location, outer surface of thylakoid, inner surface of thylakoid, can be seen in both the grana and stroma lamellae, while photosystem 2 only in grana lamellae. Now, involved in P700 involved in both the cyclic and non-cyclic flow of electrons, while P680 only in non-cyclic flow of electrons. Water connection. PS1 not connected with the splitting of water, photolysis, while PS2 is concerned with the splitting of water. Now, thylakoids. Non-oppressed part of grana and stroma thylakoids, non-oppressed. While pigment system 2, it is oppressed part of grana thylakoid. Likewise, many more are there, at least 4 or 5 differences between pigment system 1 and 2. If we can remember, that is good. Now comes assimilatory power. So what is the meaning of the term assimilation? There was a chapter called malnutrition. Presently, it is not there, where there is a heading, nitrogen assimilation. Assimilation generally means taking into the body. And in photosynthesis, assimilatory powers are referring to mainly ATP and NADPH. So energy produced in the form of ATP and NADPH2. And these two will be produced during photochemical phase or light reaction of photosynthesis. And the term assimilatory power was given by a famous scientist, Arnon. He also discovered photophosphorylation around mid-1950s. Arnon, assimilatory power and photophosphorylation. I think that idea, that is why I told you when we discuss historical events, certain names will be coming when we proceed. So another name came, that is Arnon. Now comes electron transport system in photosynthesis. ETS or ETC, we already studied in respiration, that is respiratory. Here it is photosynthesis, connected with the scientist Hill. And what is ETC or ETS? Anywhere let it be. Basically, it is a series of electron carriers will carry electrons in a downhill pathway. Why it is downhill pathway? So release of energy will be there. And that is used to develop an electrochemical proton gradient, by which ATP can be synthesized. Chemiosmotic theory you already know. That is ETS or ETC. Now comes the term photophosphorylation. Photo means light. Phosphorylation, we already studied in respiration, adding of phosphate. So counter is dephosphorylation, removal of phosphate. And in photophosphorylation, ATP is produced in connection with the light. That is why photorespiration will be calling it as oxidative phosphorylation, oxidation reduction. Here it is photo, light. And photophosphorylation basically two types. One is cyclic and other is non-cyclic photophosphorylation. I think this page is clear. So we can proceed to next page. Different diagrams are available. I got almost 3 editions of NCERT and the diagrams will be different like this. Present NCERT diagram, you can take care. Now first, what is the meaning of the term cyclic photophosphorylation? Cyclic means something that is going from a place and coming back to same place. That is why the term cyclic. So during ETS, electron transport system, electrons get ejected from PS1, that is P700 wavelength absorbing chlorophyll, PS1. Come back to the original place after absorption by P700 or chlorophyll molecules. Then come back to the original place after absorption excitement of electrons. During this travel, ATP will be also produced. And this is performed by PS1 since it is concerned with the P700. And cyclic, it is PS1, independently it is able to do. Diagrams you can find. First diagram, one edition NCERT, second diagram, another edition NCERT, but from that diagrams it is not clear. But these two can be labeling question and even theory they can ask. Little bit another way, I drawn a diagram that is starting with the P700. Absorbed energy, electron get excited, received by pyridoxine reducing substance FRS, then to pyridoxine, then it is transferred to plastoquinone, then to plastocyanin, again coming back to the place from where it has been ejected out, P700. That is why cyclic pathway. Energy produced. And energy production takes place between cytochrome B6F, sufficient electron volt difference, ATP will be produced. This is how that pathway takes place and energy will be produced. So that pathway, more expanded forms are there, but NCERT mainly depends on the diagram shown. More expanded diagrams are there. And little bit complicated diagrams also available for this type of charts. So simple way, P700, electron get excited, pyridoxine reducing substance, pyridoxine, plastoquinone, plastocyanin, again coming back to P700, ATP will be produced between cytochrome B6 and F with the sufficient electron volt difference. Now the chemicals involved, FRS. FRS is pyridoxine reducing substance. FT, it is pyridoxine, is basically protein and may contain iron sulfur atoms arranged as iron sulfur clusters. Then plastoquinone is a terpenoid. That is terpenoid quinone chemical. And terpenoids are somewhat lipid in nature. Now comes plastocyanin. It is a copper containing protein. And we can find only product formed in cyclic is ATP, connected with the cytochrome B6 and cytochrome F. Primary electron donor is P700 in cyclic. Final electron acceptor is same place, electron will come back, so it is cyclic, it is P700. That is regarding cyclic pathway. Now comes non-cyclic pathway. Again, 3 sets of diagrams are there, older editions of NCERT, but just put only one. And I expanded that diagram in an easy manner. Again, there are more compounds or more confusions, or we can expand it too much, but no need. In non-cyclic photophosphorylation, we can find both the photosystem 1. Here we can find photosystem 1 and photosystem 2 are involved. They work in a serial manner. Starts with the PS2, then PS1. And these two photosystems are interlinked. They connected through well-known electron transport system. And what are the products of non-cyclic photophosphorylation? ATP, NADPH2 or NADPH assimilatory powers, and there is release of oxygen. I think idea clear. Now, what is the pathway? Here you can find, starts with the PS2, P680. Electron first absorbed by phyophytin. That is chlorophyll without magnesium ion, phyophytin. From there, it is passed into quinone, then plastoquinone, then plastocyanin. And from there, it is not coming back to PS2, not coming back to PS2, it is going to PS1. That is how it becomes non-cyclic. PS1, it is concerned with the P700. Hydrogen jumps, so electron get excited. It is received by FRS, ferredoxin reducing substance. Then into ferredoxin. From there, it is NADP plus. Then you can find water also gets splitted. Protons, electrons, oxygen will be released. And this NADP plus 2 electron, in the presence of the enzyme ferredoxin NADP reductase, H will be added. It becomes 2 NADPH plus H. NADPH. In photosynthesis, it is DPHP. And that is how the non-cyclic takes place. Splitting of water also will be conveniently taking place at the same time. And we can find protons, oxygen, and electron will be released. And that is regarding non-cyclic photophosphorylation. Now comes connected with the splitting of water. So we can find here there is photolysis in non-cyclic. Splitting of water will take place and it is called Hill's reaction, otherwise called photolysis. Hill's reagent is DCPIP, dichlorophenol indophenol. And this DCPIP is a dark blue solution, changes to lighter when get reduced. So protons, H+. Water splitting complex, photolysis, that complex is associated with the photosystem 2, and which is physically located on the inner side of the membrane of the thylakoid. It's a famous statement from NCERT, usual question. And this water splitting complex is same as OEC. OEC means oxygen evolving complex. And the products of non-cyclic photophosphorylation include ATP, NADPH. These two are assimilatory powers, and there will be release of oxygen. Now comes a comparison or differences between cyclic photophosphorylation and non-cyclic photophosphorylation, like photosystem 1 and 2. Everything written there. Many are there. 6, 7 items already came for exam theory and comparatively more important that way I put it. Cyclic, external source of electron not required because same electron will come back. Other side, that process requires external source of electron, donor. Now, cyclic, performed by photosystem 1 independently. Non-cyclic, both the photosystems are needed, PS1 and PS2. Then, cyclic, third one, no photolysis of water. Non-cyclic, photolysis of water will be there. Cyclic, no oxygen released. Release of oxygen is available. Cyclic, only ATP produced. Non-cyclic, ATP, NADPH, and even oxygen released. Cyclic, it is exclusively seen in bacteria. Non-cyclic, it is connected with the carbon dioxide fixation in all plants. Now, seventh one, it operates under, that is important, low light intensity, low carbon dioxide, and anaerobic condition, cyclic. So non-cyclic will be just opposite. Optimal light intensity, presence of carbon dioxide, aerobic condition. Just and on, it's a famous question. Now, cyclic, it is seen in stroma lamellae membrane. Mostly we can find that. While non-cyclic, it occurs in granule thylakoid, grana. Very predominant exam question for theory entrance. Now, as usual, 5 theory question, 5 NEET model question with the key given. So we can just pass through what all things we studied or we passed through in today's episode. So we started with the photosynthesis, third one, where 9 items are discussed, starting with the photo, then photo prefix, 5 terms, then photosystem. Before it, 2 parts of photosynthesis, light and dark reaction, then photosystem of pigment system or PS or PSU. After that, we proceeded through light harvesting complex, PS1 reaction center, light harvesting complex, which contains LHP and protein. Same with the PS2. And PS1 and PS2 has got many differences. And that list has been given there, differences between pigment system 1 and pigment system 2. Many we can observe in cyclic and non-cyclic photophosphorylation also. Then we passed into assimilatory power, meaning, and the term given, identified by or given by Arnon. Arnon also discussed photophosphorylation. Then ETC in photosynthesis connected with the Hill, of which photophosphorylation 2 types, cyclic, non-cyclic, and their pathway, and what all products will be formed in cyclic, and what all will be formed in non-cyclic. Non-cyclic is also called as, that I forget to say in the first one, Z pathway. It is there. Z pathway because it appears like a letter Z this way. Letter Z it appears, but originally when we look on it, we can find it is like N here. But scientists say it is Z pathway. Connected with it, water splitting, it is Z protein. And Z pathway is otherwise called Z scheme. Then we proceed into DCPIP. What is the idea? Then products of non-cyclic ATP, NADPH as well as oxygen. Then differences between cyclic and non-cyclic. After that, 5 theory, 5 NEET type question with the key. That is regarding today's episode, Photosynthesis 3. If you like it, episode, please subscribe all, like it, share it, comment on it. We can meet later. Thank you.